Welcome to the part 3 of the lecture. In the previous parts, we reviewed basic methods of phylogenetic analysis, generated and analyzed our model sequences using the MEGAS program. In this part, we are going to apply these skills to a challenging question of the origin of SARS-CoV-2. We start with a brief introduction to the COVID-19 disease and its causative agent, beta coronavirus SARS-CoV-2. We select a key protein, so-called spike protein, from the virus, build and analyze its phylogenetic trees of relatedness to a representative set of known alpha and beta coronaviruses and compare some key coding RNA sequence features. This will form the basis of our discussion on how probable is it that SARS-CoV-2 is lab-made.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gather just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane.
S protein. SARS-CoV-2 makes use of a densely glycosylated spike, or S protein, to gain entry into host cells. The spike protein is composed of 3 identical chains that together form a complex with the small domain inside the virus, a membrane-spanning segment in the middle, and a large ectodomain that extends outwards from the virus. In addition, the spike is a glycoprotein. The ectodomain is covered with sugar chains that help to mask the virus from the immune system. The structures of SARS-CoV-1 and SARS-CoV-2 spikes include only the ectodomain, and as you can see, they are very similar. Each chain is synthesized in one piece, but then is clipped by cellular proteases into 2 functional pieces. This is a very important feature to which we come back later. The outer S1 fragment, colored green, binds to cellular receptors, and the S2 fragment, colored cyan, directs fusion of the virus within the cell. Recent structures have revealed that the spikes of SARS-CoV-1 and 2 are quite flexible. In these structures, the receptor binding domains are seen in different conformations. Often, just one domain is extended upwards, but sometimes they are all tucked down or several are extended. The extended conformation is needed to bind to receptors. This flexibility is a great advantage to the virus. Spike protein binds to ACE2, angiotensin converting enzyme 2, on the surface of cells. ACE2 is an enzyme that activates angiotensin, a peptide hormone involved in control of blood pressure. ACE2 is found on lung, heart, kidney, and intestinal cells, making these cells the target for infection by the virus. In the complex of ACE2 with the receptor binding domain of the SARS-CoV-2 spike protein, ACE2 is also bound to the amino acid transporter B081. SARS-CoV-2 S protein shares 98% sequence identity with the S protein from the bat coronavirus RAD-G13. The most notable variation arises from a 12 nucleotide insertion in the S1-S2 protease cleavage site that results in a 4 amino acid furin recognition site in SARS-CoV-2. It is shown in red in the structure. Here, in the structure, only one protomer out of 3 is colored to highlight its trimeric composition. The receptor binding domain is shown in green, and it is in the down inactive conformation. The N-terminal domain is highlighted in blue, and the glycosylation sugars are yellow. Notably, amino acid insertions that create a polybasic furin site in a related position in hemagglutinin proteins are often found in highly virulent avian and human influenza viruses. This insertion of the new furin cleavage site increases virulence of SARS-CoV-2 almost 2 orders of magnitude as compared with SARS-CoV-1. The S1-S2 junction is in a highly conserved, albeit in a disordered, solvent-exposed loop, only partially resolved in the structure. In addition to this insertion of residues in the S1-S2 junction, only 29 variant residues exist between SARS-CoV-2 S protein and RAD-G13 S protein, with 17 of these positions mapping to the single receptor binding domain. Alright, after loading my S protein encoded RNA of 270 clusters plus 22 alpha coronaviruses. So we obtain an alignment after alignment and loading into the mega. So we obtain the following data set. So here we have the names, and the names look like that. So first B means beta, so this means beta coronavirus. Then here we have NICB accession number, and here there's a textual description. What is that? So in particular here, Wuhan seafood market pneumonia virus. And the last is the number, this number, or all of these numbers. This is alignment score of a given S protein from this virus to the SARS-CoV-1. So the higher the number, the better the alignment. So the maximum alignment should be about this 2300, something like that. So the numbers less than that means that alignment is less complete, so there are like changes, mutations, and so on. So here this is protein encoding sequence, but RNA. So therefore it's broken down into the triplets. So here is the one triplet, and this is the next. Then this is next triplet, and inside of these triplets we have consensus sequence. So if, let's say, the dashed line means that at that position in the other sequences we have same number, like same letter T here. So we can also scroll to the right and see there's a quite a variation all around the place. So this we will use for the phylogenetic reconstruction, this data set. So how we can go on with this? We go straight to the phylogeny. So since the data set is big, we have to choose fast methods. The fastest method out of these 5 would be then the neighbor joining trees. And so when we run it, we are going to bootstrap our phylogeny. So we choose 100 number of replications, so not 500 as we used before, because the data set is big and we wanted to complete it in a reasonable time. So then we just execute it. After like about 3 to 5 minutes, we'll get these results. Okay, let's explore. First of all, what do we have here? So in the middle here we have tree, phylogenetic tree, which is neighbor joining, calculated by neighbor joining methods. And then here we have bootstrap numbers. So anything above 50 gives us some confidence that the reconstruction is correct. So here the numbers are 100%, so here is coming to 50. So then on the right side, here we see the names of S proteins. So alpha coronavirus, then accession number and ICB accession number, then the name, yep, and then the alignment score. So what we have here, first of all, we have alpha coronaviruses. They should form compact outgroup. Let's see whether they do form outgroup. Yeah, so they, see, this is the root of this viruses. So, and here they form a compact group. Let me compress this group so that we see that it's compact. So I click here on the node, and then I choose compaction style. Yeah. So, all right. So here we have bootstrapping 100%. So we've got alpha coronaviruses as a group, and this is very convenient outgroup for us to root the tree. So we basically root the tree, and we don't need to do it right now because it's already rooted. So you see, this reaches this branch all the way to the left side. So it's indicating that grouping more or less worked correctly. So now let's move on. And then we have a green plate in here. And what is inside the green? So we have SARS-1 coronaviruses. So we have a variety of them. And so here bootstrapping shows a low number, but these are all related coronaviruses. It means that if they are very closely related, they're just simply taken from different places and so on. So then this particular branching point might be not correct, but all in all they form a compact group. So what we have here in the SARS-1, so we have civets, we have SARS coronavirus, SARS-1. So this is civets, and then as we know that in SARS-1 case the civets were the intermediate hosts and not the high bootstrapping numbers, so they're grouped together with the SARS coronaviruses. So, and then that's why the civets were established as intermediate host for SARS-CoV-1. Again, the civet here. So let's compact this group. Okay, so we compacted it to SARS-CoV-1. Now we have to scroll up and find, these are bats now, various animals and so on. So we're looking for the SARS-CoV-2. And here this is in purple, SARS-CoV-2 group, like this. So what do we have here in this group? We have bat coronaviruses. So we have Wuhan seafood market pneumonia virus. So the human one, closely related but sampled later another version of the same virus. Then we have pangolins. So here is the group of pangolins. You see how high the number, the alignment score is. So of all of these pangolins. So really the pangolins are closely related. But, and this is the most interesting, so the famous rat G13 is grouped together with the human coronaviruses. And this is no surprise because it's 98% identical to the human coronaviruses, but that one's from the bats. So sampled in 2013 and kept in the lab since then in the Wuhan Institute. So we have now three important viruses in the SARS-CoV-2 group. So pangolins, one more bat, but more distant. So, but we take a note of this. And then we have Wuhan coronavirus. So let us look at the furin cleavage site across these coronaviruses, so which form SARS-CoV-2 group. Here I selected SARS-CoV-2 group of the viruses. Let's consider, take a closer look. So what is that? So here we have 5 of them from the alignment. So the first would be Wuhan seafood market pneumonia virus, so SARS-CoV-2 in here. Then we've got this famous, rather infamous rat TG13. So rat G13, bat coronavirus. So the closest relative to the SARS-CoV-2 in terms of the S protein. Then they have SARS-1 sequence, then pangolins. And we have also one more bat SARS coronavirus. So now let's take a look. Okay, we see here pretty strong conservation of all the positions in the protein amino acid sequence here. So sporadically here and there we see like mutations, 1 or 2, but most of the time it's the sequences are quite conserved. But what if we look at the corresponding DNA sequences? We see that there's quite a variation in here. So how this can be? The answer is like this. So genetic code is redundant. It means that we can change triplets, but they will still encode the same amino acid. So, but looking, and if let's say we change a nucleotide, but it doesn't result into the amino acid change, it's called synonymous substitution. However, if let's say nucleotide is changed, but the amino acid is also changed, it's called non-synonymous substitution. So comparing synonymous versus non-synonymous substitutions, we might tell how strong is the selection. So if selection is purifying, very strongly purifying selection, so then we will see alterations here and mutations here in nucleotide sequence and not much of the protein sequence. So like here. Now let's scroll. So we observe here quite strong purifying selection. Let's scroll to the furin binding site. Here it is. So we observe this insertion PRRA only in the SARS-CoV-2 S protein. In none of the related SARS-CoV-2 viruses, so like bat, rat TG, rat G13, or SARS-1, this one, so or pangolins, or the bat, the SARS-1 from bat, so they don't have this insertion. though this region, as you can see, it's very conserved as well. So if we flip to the DNA sequences and then find this insert, 12 nucleotide insert in here, that 12 nucleotides insert in here, resulting into the 4 amino acids insert. We can see quite a bit of variations in nucleotide positions. These are all synonymous substitutions. So because purifying selection doesn't prevent nucleotide substitutions in here, only amino acid substitutions, right? So therefore we see quite a variation around here as expected. So in quantitative terms, so the number of synonymous versus non-synonymous substitutions, so synonymous versus non-synonymous would be then in the order of 0.1, so which indicates very strong selection in here. And in none of the related species we observed this. So it means that it's a very important area in the protein in here. So whether this can arise by chance, it's very unlikely, because here we have a very strong purifying selection acting on all of these areas around. So, and then how this sequence can evolve into this sequence, unless we assume that it was just artificially inserted, just this part, increasing twofold virulence. In the latest discussion regarding the origin of furin cleavage site in here, so what is the origin of the furin cleavage site? They state that indeed, so Wrapp G13 doesn't have this site, and that's what we just discussed a minute ago. And then they said, no, do any of the other bats, SARS-CoV or pangolins, have been isolated having this site? And that was the smoking gun of the engineering. But they say, however, recently a new isolated SARS-CoV-2, this one, which is not available through NCBI as yet, was shown to contain a polybasic amino acid insertion in the spike glycoprotein. So, and from that it was concluded, so that is SARS-CoV-2 arose by recombination among bat viruses in nature. It looks like it's disproving the entire smoking gun hypothesis. However, if we look at this paper, so where this novel bat coronavirus was described, it's actually, first of all, the paper is coming from the Wuhan lab, from this famous lady director of this institute. So, but even if, let's say, we accept that, so we can look at the alignment in here. Here it is. So let me pull it up here. And what we observe here is this newly discovered virus. And what we see here, that these few amino acids are inserted at that position, but they are not of polybasic character. So this is arginine, or proline, arginine, and these two arginines, they form furin cleavage site. So here there's a proline inserted, but here there's a gap, and then alanine inserted in that position. This is a not polybasic furin cleavage site. And you can see there's quite a number of the missing residues around here and around that position. So therefore, it doesn't disprove actually anything. This is quite interesting debate which is going on right now in the science, and I would like to bring this to your attention. And yet another interesting observation made that tried the same data set which we analyzed, exactly the same. So what they found extreme genomic deficiency in SARS-CoV-2. So as an evasion of the host antiviral. So what they say, it said here that put it like this. So what is this CpG deficiency? So CG increases stability in the RNA or DNA structures. So therefore, like genes, the areas which encode some proteins, so usually enriched in CG. This is one of the markers how genes are selected. However, in the SARS-CoV-2, extremely low CpG frequency is found. So here it is. Yeah. So SARS-CoV-2 has the most extreme deficiency in all beta coronaviruses. And it suggests that SARS-CoV-2 may have evolved in a new host or new tissues, actually being transferred from tissue to tissue and so on. So that would result into the high CpG deficiency in the virus as stated in this paper. So now if we look at the graphical representation of this, so the extreme positioning of this is amazing. So in comparison to the other coronaviruses, for example, human infecting HQ1, so it can code, doesn't show quite low deficiency in here, or so the other viruses. So these two, Rat G13 and SARS-CoV-2, take very separate positions in terms of the CpG deficiency. So it seems that this virus and this virus and derivative of it, this one, were passed through multiple tissues. So how this can be? So if it's just host in bat, this is bad species. But if it kept in the laboratory and was passed through the several generations, through the several tissue cell types, that might happen. Exactly this might happen. This concludes our excursion to the origin of SARS-CoV-2 coronavirus using methods of the phylogenetic analysis. Of course, our objective was not to prove or disprove the artificial origin of it. The objective was to learn how to use the phylogenetic analysis methods to create a basis for discussion of this exceptionally difficult and important question. In the following application exercise, we are going to summarize arguments for and against the lab-made or natural reservoir hypothesis, which perhaps will prime us for new scientific discoveries in this field. However, larger implications of this debate for future human evolution still hold true. If SARS-CoV-2 is a natural virus, we may be in a good position to prevent future epidemics through wildlife protection and conservation. If it's lab-made, this certainly heralds a new era of disruptive biotechnological developments, of which we shall stay aware.